Okay, let's move on to another major type of membrane lipid. So these are lipids that do not use glycerol. We just spent a bunch of time talking about glycerophospholipids. Those were all lipids that at least had glycerol as its backbone and had a phosphate group on them. These membrane lipids utilize a different type of backbone, so no glycerol, and they are called sphingolipids. So again, the backbone of a sphingolipid is not glycerol. We're going to be attaching all of our groups onto a different backbone. Instead, the backbone is a long chain alcohol that is called sphingosine. Sphingosine is just a unique molecule that is utilized as the backbone for sphingolipids. Now, these fatty acids, instead of attaching to glycerol, which remember attaches via an ester link, is going to attach to sphingosine via an amide linkage. So we're attaching with a nitrogen bond instead of an oxygen bond. Now, sphingosines also are going to have an attached fatty acid, and they can have different head groups, polar head groups, that give them unique variety. These head groups, instead of being attached by a phosphate, like in a glycerophosphate, are attached by a either glycosidic bond or a phosphodiester linkage. Again, we'll look at that. There are some type of sugar-containing sphingolipids. These are called glycosphingolipids. So this is just a special branch of sphingolipid. And these are found largely on the outer portion of membranes. So let's look again at the basic structure of a sphingolipid and how the varieties of them that can exist based on the head group portion. So you can see in pink here, that is our sphingosine. This is the backbone. That whole long molecule is the backbone of a sphingolipid. And again, the key here is that the nitrogen on the sphingosine attaches to the carbonyl of a fatty acid. So it makes an amide bond instead of an ester bond. Remember, an amide is when you have a carbonyl attached to a nitrogen or an amine. Now, you can also see that at the end of our sphingosine, where we have this O group, is where we attach our head group substituent. A very, very basic sphingolipid that just has a hydrogen as its head group is called a ceramide. And again, these ceramides we can always change based on what fatty acid is attached to it, but the general name for them is a ceramide. You can also attach to this whole head group a piece like a phosphocholine. So putting a phosphate group on and choline group on to that whole end of a sphingosine gives you a special type of sphingolipid called sphingomyelin. Sphingomyelin is very important and is utilized often in the brain. Again, you can attach other things here like sugars. These are in some cases called glucocerebrosides. Again, very important in the brain when you have these sphingolipids with sugars on them. Sphingomyelin, which again is our sphingolipid that has some sphingosine group, some fatty acid, doesn't matter what fatty acid, and a phosphocholine head group, is abundant in what is called the myelin sheath of the brain. This is the wrapping that, these are a lipid wrapping that occurs around nerve cells. It's kind of like if you were to look at a wire, the waxy coating around the actual internal metal wire helps protect the wire, and it actually helps allow the electricity to conduct better. So the coating around wires helps with the conduction of the wire itself. In our brain, we have the same thing. We have nerve cells that conduct electricity and a myelin sheath that has to be insulating and helps the electricity conduct. And this wraps around the nerve cell. These myelin sheaths, again, are mainly made up of lipids that are very insulating. And in particular, sphingomyelin is one of the major components of this type of coating, which again is called the myelin sheath. Now, note again that sphingomyelin is structurally similar to a phosphatidylcholine, a PC group. Remember, phosphatidylcholine uses glycerol as its backbone. It has 2 fatty acids and a phosphocholine head group. Sphingomyelin, structurally very similar, except remember that this whole portion is our sphingosine. But it does resemble a fatty acid group, right? It has very large hydrocarbon tail, similar to a fatty acid group. Now, another type of glycerosphingosine is very important in things like blood groups. These are going to be sphingolipids that utilize a sugar on them. It looks like this. So this is a type of sphingolipid. We're still talking about sphingolipids. It has a sphingosine backbone. There's a fatty acid attached to it, but its head group is a sugar. Now, blood groups, having a blood of A, B, or O is actually determined based on the number of and the type of sugars, head groups that are on a glycerosphingolipid. So glycerosphingolipids, sugar sphingolipids are responsible for determining your blood type. This structure, the actual structure of the sugar, is determined by a particular enzyme, a glycosyltransferase. Essentially, if you have no active glycosyltransferase, you'll have an O antigen. If you have a glycosyltransferase that transfers a sugar called an N-acetylgalactosamine, you'll have A group. And if you have instead a glycosyltransferase that transfers on a galactose, you'll have a B group. So I'll show you what that looks like. Here is the different types of lipids that could be expressed on your blood cells. They are glycosphingolipids. So they have a sphingosine backbone. They have a fatty acid attached to them, but they have a different string of sugars. You can see that right here is the same between all 3 of the potential blood types. But what is added onto the end of this chain is going to change which expression you have. So if you add nothing onto the chain, you have an O blood type, an O antigen. Again, if you add this N-acetylgalactosamine, this is a special type of sugar, you would have an A antigen. And if you added a galactose on the end, you would have a B antigen, B blood type. Now, there are other sugars that are involved with this chain, but notice that they are the same amongst all 3 of the antigens. That whole portion of 5 sugars in the beginning stays the same throughout O, A, and B. It's only that last position that is the little change that would make you an O, A, or B type blood type. And again, these are all on glycosphingolipids.